Yes. Even if nobody can show up, I understand. It's just that's when I have time. Like, I was thinking, I was like, well, I'll just pre-record it and just post it. I'm like, well, if I'm gonna pre-record it, why don't I just Zoom it while I'm doing that? Like, it didn't make any sense not to, so that's why it is that way. Right, probably go ahead and get started so we don't lag too far behind. So does anybody have any questions about anything before we do the Kahoot? All right, what is the term that refers to an organism's total sum of genetic material? All right, so most of you guys got this one. It's going to be the genome. So the genome is going to include any chromosomes that are in there, and the different versions of genes and whatever. So that would also include in plasmids if you're like a bacteria with extra plasmids that had extra genes on them, or like we have in our mitochondria, they have their own chromosome there, but that's included in our genome for we need that to survive. So full sets of genome, total genetic info. All right, next question. We have the physical expression of a genotype. This is called a phenotype. I'm glad most of us remember that word, and we know the difference between those. Remember in that picture, it was showing us like big R, big R, big R, little r, and whatever. Those had equally expressed alleles, which are different versions of genes. And we didn't really talk about alleles, but if you have two dominants, how do you choose which one is which? Sometimes both are expressed. So instead of, white, which might be dominant, and then the red flower, which was dominant, and then having both, you get pink. Sometimes that happens too. So we didn't get into that too much, but anyways, no matter what, the red, color would be the phenotype, or the pink color would be the phenotype, not the genotype. The three parts to a DNA nucleotide are, read your choices. All right, so most of us getting this one as well. It's going to be the sugar, which is a pentose sugar of some kind. When we're talking about nucleic acids, but when we're talking about specifically DNA, we're talking about deoxyribose. Remember, RNA is going to have ribose as the sugar. Those are sugars. Ose at the end, sugar. Okay. In our backbone, we're also going to have that phosphate. If we remember talking about how our DNA is made up, it's going to be sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate along the backbone, and which orientation of that sugar determines, the direction of that specific strand. Just the one strand, not the double helix. Anyways, and then the nitrogenous base is what will bind off of the sugar and be the A, T, C, or G, and it can hydrogen bond across to the other one, respectively. I didn't know that was on. All right, next. That's a lot of review all in one thing, but there we go. I know that chapter is a lot. So I want to make sure we kind of cover it well. All right, DNA anti-parallel strands of nucleotides are linked by, so I'm talking about the strands of the nucleotide. This strand of nucleotide, that strand of nucleotide, how are they linked together? All right, so this is getting at the hydrogen bonds that are happening here. You have your one strand going 5' to 3' in one direction. I just do it like arms. It doesn't really explain very well, but have one strand going from, like, this would be 5' end to 3' end. The other strand is the opposite orientation, which is why it's called anti-parallel. These are two strands that we're talking about. We've got our different nucleotide bases here, so our nitrogenous bases coming off here, making this A, T, C, or G, or whatever. And then them, like if this was A and this was T down here, hydrogen bonding between the whole way down. So that's where that comes back into play, where we learned about that in the very beginning. These two strands that are anti-parallel to one another, each one is made up of nucleotides, and running the strand as a whole, running opposite directions from one another, and they're only held together by hydrogen bonds. Pretty neat stuff. The nucleotides themselves next to each other, though, covalent bonds, right? So true or false, DNA polymerase builds DNA in the 5' to 3' direction. Is that the correct direction? And is that the correct enzyme? All right, so this is true. I feel like this is pretty straightforward because I keep saying over and over again, 5' to 3'. So hopefully that'll be a gimme when you encounter it on the exams. Which enzyme connects the Okazaki fragments that are formed during DNA replication? This does involve understanding a little bit about that DNA replication process. So that enzyme, we briefly mentioned it, is DNA ligase. And we just said, hey, they're joined back together by DNA ligase. And that's kind of how we left it. But the concept of how DNA ligase works and what it does for putting the strand together to make it whole again is going to be extremely important in the chapter we're going to get into today. So we can't do any genetic engineering without DNA ligase. It just doesn't work. Because if you have your Okazaki strand that was, made in the other direction, and you have it in pieces, and they're not sealed together with those covalent bonds that should be there holding it together, then the next time that you go to make a new cell, they're in pieces. The next time you open that gene up to make mRNA, it's in pieces. So it isn't useful to the cell that way. So literally cannot survive if those are left in pieces. They have to be sealed back together. And remember, the reason we have those pieces is because we can only add those nucleotides in that 5' to 3' direction. So as you're opening up your replication fork, gradually one side can just crank on into the fork and keep on trucking, and the other side has to make it in pieces as the fork is opened up on its side. So that gives us those pieces. Those fragments are called Okazaki fragments. And if they're fragments, we have to seal them back together. DNA ligase is going to ligate those pieces together by joining the nucleotide next to each other in those fragments by their normal covalent bond. So they're helping regenerate. We call it a phosphodiester bond, but I'm not necessarily expect you to pick that out of a lineup, okay? But that covalent bond there. So that the DNA can be functional. Okay. All right, next question. During transcription, what's going to happen? And this is an image of transcription. All right, so even if we weren't really sure what the term meant, which I'm hoping you guys are getting eased into those terms a little bit by this point, but transcription is the RNA to DNA process. And even if you weren't sure about it, you could have looked at that picture, and the pictures don't always line up, I'll be honest, but this time it did, and I told you that it did. And so you should have been able to see we're opening up that DNA and getting a single stranded thing out of it, trailing out of it. And it was clearly still nucleotide in the language. So that had to be RNA. Anytime, by the way, anytime you make any kind of RNA, even if it's ribosomal RNA, because that had to come from DNA as well. There's genes for that. Or transfer RNA, those little T guys that are going to bring us the amino acids. Those all had to come from DNA genes. So anytime you're making those from the DNA genes, it's still called transcription. Okay, it's not only going to be mRNA. It's just we're more interested in that part of the process when we're talking about it. I'm gonna have to shut that door, so I can't deal. A codon is 3 bases of what? It's been like this since the summer. All right, okay. A codon is 3 bases. So a base, anytime we're talking about a base, we're talking about the A, T, C, G code of whatever. And remember we talked about size. I don't know if we did talk about size. We did talk about size, didn't we, in the last chapter? Or is that this chapter? Did we get to it yet? How we say how big a, okay, we'll talk about it in this chapter then. Anyway, so when we talk about something like A, T, C, or G, you could say that is 1 base. A is 1 base, T is a base, and whatever, in the code itself. Like obviously they're nitrogenous bases, but I'm talking about in the code itself, you can still say those are bases. I'm saying it for a reason, I promise, but it'll make more sense later. So anytime we are talking about those three bases, the codons for the mRNA is talking about the ATC would be DNA, which would be like AUC in mRNA, and AUC codes for serine. Well, that AUC in the mRNA is what's going to tell the ribosome which tRNA we need. So it's going to match up with its opposite, right? Its anticodon. That codon is number one of the most important things you need to understand what it is, what its term is, and all that when you're trying to understand this stuff going forward, the genetic side of it. Once we get into other topics, it probably isn't going to come up again. But yes, because we're going to get into, like, if this chapter was difficult and you're having a hard time understanding the major topics in this chapter, chapter 8 is going to be rough, like I said. So I'm about to talk about procedures that we do with DNA that are based off of your knowledge that you should have about the DNA from the previous chapter. I mean, yeah, the book expects you to learn it from one chapter prior as well, which, you know, is tricky. But anyways, mRNA, we can divide it into those three nucleotide, and each three nucleotide segment is called a codon on the mRNA. And that codon codes for an amino acid, but it itself is, you know, going to be mRNA. If that makes sense. So. Cool. All right. In what kind of operons? We have repressor proteins that bind to operators unless a molecule that changes that repressor shape is present. Fancy long word way. We're phrasing this too. We're talking about a gene that isn't turned on until we get the signal to turn on. That's all that's happening here. All right, so again, in our story here, in this really wordy statement, which I'm sure once we take time to really go through the terminology and be more familiar with it, would have made more sense. But, and whatever kind of operon, so it's asking us what kind of operon has a repressor protein bound to its operator, which is just where they bind. Okay, repressor protein doing its thing, unless we have something to cause it to come off, basically is what it's saying. So that has to be inducible. So it's being repressed. So the only way, next thing you can do is induce it. If it's already on, the only thing that you can do is repress it, right? So keep that in mind. Sometimes working through the words or trying to figure out like how the words referring to something can be harder than the concept, honestly, because the concept of it really isn't bad when you explain it that way. Okay. Missense mutations result in okay, when you have a missense mutation, here we have one amino acid being changed into another. It's still an amino acid, but it's the wrong one from what it should have been. Remember, a non-sense mutation is going to be if you change your nucleotides and now it gives you a stop. That would be bad. And then silent mutation is you change your nucleotides and it still gives you the same amino acid. It's just a different way to code for it. So, and of course, back mutations, which I don't really talk about a lot, is just reverting back to what would have been like the original code. Something you typically see more in like bacteria and viruses than you do in eukaryotes. At least it's hard to study it in eukaryotes. Good job, guys. That's not a cursor. There it is. All right. So good work. Anybody have any questions? Again, once again. All right, so we're going to jump into chapter 8. This is our genetic engineering chapter. All right, recombinant. What is this thing that is like popped up down here? Let's see, where's my cursor? Get out of here. You also get out of here. Okay. Recombinant DNA technology is a technology that we have developed based on mixing up genes between organisms, basically, is what we're talking about. The tools that we have developed based on that technology have led us to things we can do to, identify people, whether it's like paternity test or who murdered so-and-so and whatever. We'll get into that today. Fixing underlying genetic mutations to treat diseases. We're going to get that into that today. And I'm not talking about looking at this in research in your little tiny cells on your little, thing, or even doing this with mice and saying, see how we changed it back from the mutation we already induced into it. No, I'm talking about in people, live people, adults. We can change their mutations back to healthy forms, literally in their cells. That's not cool yet, y'all. It should be. So it should be. And then talking about utilizing CRISPR technology to repair genetic mutations in a heritable way. We're talking about heritable. That means not just changing your genes in you so that you are fixed, but also changing you so that if you have children after that point, your children would also not be carrying that disease, which is like a great goal. I'm sure some of you have heard about CRISPR. We're going to talk about that a little bit today, not too much. I don't want people to get disillusioned into thinking like it is this massive, miracle to save us all. Science is not effective at using it at all right now. So just say it. If somebody told you otherwise, they is lying. All right. We can get it to work in mice, but it just seems to cause more problems sometimes than it does help. And we'll talk about why that is. So a lot of what we're going to be talking about today is going to be referring back to what we understand about DNA molecules. And DNA, again, double helix. There's two sides to it going in opposite directions, held together by those hydrogen bonds, right? Those bases are held together by hydrogen bonds. Weak bonds. And we've kind of already discussed how if you had a hydrogen bond in a structure of any kind, whether it's DNA or protein or anywhere, if you're introducing excess H plus or proton ions via an acid, for example, right? Decreasing that pH, increasing the H plus concentration, right? Then that could affect the ability of these two strands to bind together, at least in portions, right? You can imagine that because it would interfere. Those H pluses could get in there and be preferential for the negative oxygens to want to bind to and could cause issues with those strands being able to sit together. I know you're looking at this and you're thinking, yeah, this is talking about heating and cooling. Yeah, heating, though, that's my whole point here. If the H pluses can do it with their pH, obviously heat, which is energy that we're introducing into this. Don't forget that heat is energy. We're putting energy into this. Of course, those weak hydrogen bonds can come apart from heat. So that makes sense. And then if you cool it back, because we know A binds to T and C to G, and based on how those things will have to match up along your entire DNA and all of your chromosomes, specifically to the other strand, they will just come back together and stick back together just automatically, because that's what chemistry says to do, which is useful. Because I'm telling you now, if we can take your cells out and separate out your DNA strands from one another, do something, and then put them back together or create new ones from the separated strands, then that gives us something we can work with in our genetic engineering methods. Okay. So cool. So we know heat can separate. This is going to be our driving concept behind probably the number one most important thing you're going to learn about in this chapter, which is PCR. If you have heard of PCR, please tell me that you have, because if you know anything about COVID, you should know that PCR is a thing, right? Because they would say, you can do the home tests, and if it's positive, it's positive. But if you don't have access to home tests, and especially in the beginning, they preferred that you go get the PCR test so that we can be sure, do you have COVID or not? We're going to talk what PCR is today and how it works. Again, we don't just use this for COVID. It is the number one diagnostic tool used in hospitals all over the world. PCR is not just for COVID, for most diseases. So super important. And it's important for obviously genetic engineering, or it wouldn't be in this chapter. All right, so the first complicated thing that we're going to get into is a word that I feel like is more complicated than it should be. Restriction endonucleases. All right. These are enzymes. So ACE, we can already say it's an enzyme, but they're enzymes that are going to cut DNA at selected positions. They only cut one of the strands, each one side of a strand, okay? So they only cut one area in one strand between the nucleotides. So nuclea, we're cutting nucleic acids between nucleosides. Endo, we're cutting within the strand, right? Not from the end, but from within the actual strand. And it's restricted to specific sites. And that's why it's kind of called restriction endonucleases. These guys were originally made by bacteria naturally in nature to help combat foreign DNA, whether that be infecting viruses, the bacteriophages, trying to insert their little DNAs into the bacteria DNAs, and they didn't want that, so they could cut it out, or by plasmids that they didn't want, or something like that. So these guys, like it says, they can break the phosphodiester bonds between the adjacent nucleotides on both strands of the DNA. And it is on both strands, but it'll only cut one strand at a time. So remember, these are your phosphodiester bonds. So the ones between the nucleotides. Okay. These guys recognize sequences of 4 to 10 base pairs. It's usually around the 6 base pair length. And it tends to be even numbers every time. So, right, we already said what they do. So the spots where they're going to be binding, let's say, are called palindromes. And if you don't know what a palindrome is, in English, a palindrome is a word that reads the same forwards as it does backwards. So one of the most common ones we know is race car, right? If you read race car forward and then read it from the back, it says race car. So that's nice. So that's a palindrome. This is similar to that, but I'm not talking about one strand is a direct palindrome, but like one strand reading the exact same as the other strand in that segment. I'll show you what it means here in a second, but anyways, we will, let me pull up a little, do this so people online can see it. If you have a strand, why are you this way? You'd think it would know I want the pen thing when I'm using the pen, but whatever. Just like this. Okay, you have one strand, and let's say that this strand reads G, G, A, T, let me make sure I get it right so I don't mess it up, C, C. Okay, so that might be one strand. And we're just looking at that one strand. If you were to fold over this at this point, fold it over in the middle towards itself, if you were to do that, what does T bind with? It binds with A. What does C bind with? G. What does C bind with? G. And that will be the case with all of these guys. I'll put this back because I erased it. All of them will be like this, but not just like this, to where they could bind back on each other if they so chose, but they will literally, if they were double stranded, exist like this. We would have G, G, A, T, C, C, and then guess what the other strand reads, y'all? G, G, A, T, C, C. That's a palindrome in DNA. You see what I'm saying? And especially when I remind you, remember, this is the 5' end and the 3' of this one, and this is the 5' end and the 3' of this one. They say the same exact thing in that spot, right? In that backbone direction. This is what we're talking about when we're referring to palindromes in DNA. So yes, they can bind if you fold them in on itself with that one strand, as well as it's reading opposite, like it reads the same thing on the other side because of that as well. That's really the reason why. So yeah, so that's the palindromes. I'm not going to put like necessarily a picture of something that says G, G, A, T, C, C. Could this be a palindrome spot where it will cut? But I feel like understanding this is This is nature. We didn't make that. This is a rule nature made. Bacteria already had this. We're just using it. We just found it and are using it for our purposes. So we have found enzymes, and I mean, there are a lot of different ones from different bacteria that will cut at different palindromic sequences of DNA. And they're all palindromic sequences like this, they'll just be different sequences. And most of them are going to cut in the following way. Make a cut here and make a cut at the same spot on the other strand. That's when I say they'll cut both strands, but not straight on through. There are some that do that, but I'm way more interested in these guys, okay? And these are the by far more commonly used ones. I've used them in my research throughout my entire life. Like this is super, super common in research. So it creates these pieces here because we cut those and we cut those. We can separate those now from one another. And then the top part, I'm glad that I drew this with squares because I'm not going to lie. I drew it like this earlier and it looked like I was drawing dicks. And it was horrible. So I'm glad we did it this way, just with squares. So anyways, the top piece there, it's hard for me to draw it on here, but I would gesture with my hand if I can. But this piece up here, top with that little G, will kind of separate out from the bottom piece here, especially if we heat it up at all. And so don't want to separate because these are not bonded together anymore by covalent bonds. So it's going to be easy to separate them. So we can separate that, but then you can see all of this is still your double-stranded DNA going on that way, and all of this is still your double-stranded DNA going on that way. So now we have these overhanging single-stranded bits, right? Why would we give a damn about this? Why would this be important? Yeah, it's going to connect to something else. Exactly. So I'll show you how it works. I can't really draw that. That's like getting real. I can't do it. But so you get this. So we'll get these overhanging little pieces that we call sticky ends in science. Okay. So they're sticky ends because we have this side just by itself, the right side, just by itself, that GATCC, that little G down there. This guy wants to bind with its complement because it doesn't like being single-stranded. And the other side wants to be bound with its complement. But it's not going to do that permanently until you introduce our good old friend, DNA ligase, right? So if you just cut your DNA and then introduce DNA ligase after that, it's just going to seal back up where it was. But if you're cutting your DNA and introducing an excess of a gene that you engineered to have the same sticky ends as this, now we're talking, right? Now we're talking. This is real life. So you can see how it would have happened if you look at the squares. It's not yeah, it was bad. It really did look exactly like that. Just the way that, yeah, my shaky little hand drew it. So anyway, so moving on, the palindromes and those sticky ends, they are staggered symmetrical cuts. They're always going to be symmetrical. So cutting at the 1 and the 1, whether it's one enzyme might have 2 or 2, but either way, it's always going to be symmetrical. And they will usually be 4 to 5 bases on each sticky strand so that you can get accurate pairing up of DNA and control it a little bit better. The more strands or the more nucleotides or bases that you're involving in trying to match things up in these processes, the more specific your result will be in the end, right? Because otherwise it could just be binding with some rando fragment of DNA that just happens to have those 2 nucleotides on it. And the portion of the DNA as well. You want it to be sure that's matching up with the right part of the DNA at the right place. So the more bases you have, the more specific it'll be, that binding. But with these sticky ends, we can kind of control that because we're creating that symmetry. And we've done it with this weird palindromic sequence. That, again, we didn't invent that part. That already existed. We're just using it. Okay, so this is how it works. And so we've got at the top, I think it was the same. I didn't mean them. I know it's a little bit different than what I wrote. Let me get this going so I can pull this up so we can see it better. Okay, so up here at the top, you don't want me to have a pen? I guess I can't have a pen when I do this. Maybe. Oh yeah, I can't have a pen when I do that. That's all right. We'll just zoom in. So we look at the top at like Eco R1, okay? You don't need to know this, but just FYI, Eco R1 is named what it is because that E represents Escherichia, the C represents coli, so this is E. coli, a natural enzyme that occurs in E. coli, and then R1 is restriction enzyme 1. It's just how they named it. But these are coming from actual bacteria. So Eco R1, this enzyme cuts at that sequence, that palindromic sequence in that exact way. Okay? You go all the way to the end and you see HAE3. That is what I was saying. It can cut with blunt ends, but it's not as useful for us typically when that happens, unless you're trying to just remove a sequence and not insert anything. But they do exist, but we're way more interested in these sticky end guys. Okay, so you can see if we cut at our little sticky end site here, down at the bottom where it has the 2 strands and it has the site of cut and it has a little scissors working, right? Where it cuts will leave you one red overhang that says CTAG, and the other blue overhang would still read in the other direction, CTAG, right? It's just other direction. They read the same thing, and they're still looking at palindromes just like they were before, but they'll also have the nucleotides on the other side that's saved. So anyway, so we cut this thing in the place on, let's say, a plasmid. Bacteria deal with a lot of plasmids. They get them, they have stuff in them, helpful genes or not helpful genes, and they're easy to move them into bacteria and out of bacteria. They're very small compared to their genome, the rest of their genome, their chromosomes. Anyways, you cut that. Also, at the same time, you have in your genetic engineering isolated the gene in humans for insulin. And you are taking the genetic sequence of that, which you can literally take somebody who's healthy, cut out their gene with by other means, and manipulating it with other means. You don't need to worry about that too much right now. Making sure you have the sticky ends on there and build nucleotides onto things. Make sure it has the sticky ends so you can cut that sample that you engineered from a healthy person in the same way and introduce it into a plasmid that you also cut the same way, and now they'll stick together. And you might be saying, why wouldn't the plasmid just like stick back to itself though? I mean, it's still the same sequence, because we're going to load that puppy up with our extra DNA. So it will prefer, because there's so much more of it, to bind with our excess of the gene that we're trying to insert. So that's how we can control that. And how would you know though, if you did that, how would you know that you got that in there correctly, first of all, that it got into your bacteria correctly, second of all, and that the bacteria are expressing it correctly, right? So that's a whole other stuff. Believe it or not, we're actually going to be talking about those too. So, okay, but cool stuff, right? We can put this into DNA. It doesn't have to be a plasmid. We can put it anywhere that can be cut by that same enzyme, right? So the downside of this, of course, is that we can only cut at palindromic sequences right now from using these discovered enzymes. So we have to have discovered an enzyme that cuts at that palindromic, sequence. So that's why we have to sometimes engineer in those sequences so that we can work with a known enzyme. Remember that, okay? So we're also restricted ourselves that way because we only have those enzymes to work with in those specific spots. Because I mean, right, insulin gene, does it come with the palindromic sequences automatically at each end? No, of course not. So you have to engineer that in. Right, restriction, fragment, length, polymorphisms. And this is one of those words that I have to like stand there and think my way through every time I say it. But these guys are, when you cut up your DNA, let's say, I take just your DNA and I cut it with Eco R1 and I just see what fragments come out. Your fragments are likely to be different from my fragments, right? So that is restriction, fragment, length, polymorphism, differences in cutting patterns to give rise to different fragment lengths, basically. So we can compare organisms. So you can see where this is going to go, right? If I just take Eco R1 and I cut my genome up with it, and then I take Eco R1, the same one, and cut your genome up with it, And then we compare the fragments. Well, if you got cut at the crime scene and your blood was here, and I can, say that this was the killer's blood because it's not the victim's blood, whatever, and say, whose DNA matches these. We don't even have to run we don't have to sequence the whole thing. We can just cut it with EcoR1 and see who matches up with it based on our suspects. And that's it. And that's what they do, right? We've seen this on like CSI and stuff like that. So that's where they get it from. It's from those enzymes. All right, so we've already introduced that concept of ligase. If you're sticking things together, you need to make it permanent. Otherwise, it's useless. So we'll seal those sticky ends back up next to the new strand that it's stuck into. That's ligase, the same exact DNA ligase that we use to seal up our Okazaki fragments. Nothing weird here, okay? We can also use reverse transcriptase, which we learned about from HIV, to take samples that we may be able to get that are only RNA and make them back into DNA. And you might be saying, well, if you can get the DNA, why wouldn't you just get the DNA and work with that? Because I'll tell you this about RNA. RNA is way more unstable because it's single-stranded. Way more unstable. It's harder to isolate. It's not as easy to work with. So generally you're gonna want to aim to use DNA. However, there's a problem. Because remember our introns. Remember the crap you don't need. The stories that I tell you about my dogs. Cut it out and get your info that you need. The stories that Dr. Shearer tells you about whatever, being in the military and stuff. Cut it out and take what you need from those courses, right? It's the same thing here. Those introns, that useless stuff, cut it out, join together the exons, the coding regions, to make that working mRNA. Now that's in eukaryotic cells. It's just reviewing what we talked about in the last chapter. So that's in eukaryotic cells. So if I'm trying to put my insulin gene into a bacterial chromosome and have it express that gene, and bacteria do not possess the machinery to cut out introns because they don't have them. So what do you do? Because if you just put the DNA in there, it's not going to be able to make your product. So what you do is you make a clone of the RNA, the mRNA. So you can go into someone's cells, their pancreas cells, isolate the RNA, go through all these processes to get the ones that are associated with insulin. The mRNA for insulin that's already been cut up by the human cells, and then reverse transcriptase it to the DNA form. That's our cloned DNA. And that's where we're getting complementary DNA made from, it is cloned, but I don't have whatever's happening here. We do have our complementary DNA. It's going to be complementary to that mRNA, and then we're going to make it into double strand so we can actually work with it like actual DNA. We have the ability to do this just by adding on nucleotides and using the right polymerases and stuff. So this part isn't actually that hard whenever you understand about reverse transcriptase. But if you didn't have that, this technology couldn't have existed without reverse transcriptase. So in a way, we have to be thankful for the fact that HIV exists, sadly, because we were able to discover this. How many lives can we save with this technology? And I'm telling you, millions, right? Anybody here know anybody that takes insulin ever? Never met, ever like have a loved one and maybe a relative or a friend or anybody. And if I were to ask you guys, do you think it's like super rare to have type 1 diabetes? You're probably like, it's really not that uncommon, right? Like you know about it, you hear about it. Insulin is there, like we have it available to them and it's cheap and it's readily available because of this. Because before this, we had to literally get it out of cow pancreases or other animals and take their pancreas. We're going to eat them anyways, but we're going to also take their pancreas and try to extract the insulin itself, which is way more expensive to deal with than just having bacteria cranking it out all the time, right? And before that, of course, you remember the story, I'm sure, because I like to remind people, before they even had the cows to extract the insulin from, before they even understood about insulin, if you had diabetes, you just died. You just didn't, nobody understood it, right? So in a way, it comes around full circle. At least we got something good out of such a horrible disease. it had to be there. We got something good out of it. And next on to CRISPR. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. Not going to have you memorize that, but most people just know it as CRISPR. It's actually going to be like a complex, and it's going to exist as, we call it Cas9 and CRISPR, the CRISPR complex or whatever you want to call it, right? So Cas9 is the enzyme, and it cuts out foreign DNA. Typically in bacteria, it would be, again, invading bacteriophages or plasmids that we don't want. So it exists in nature. And these are going to recognize these very short lengths of DNA that have repeating nucleotides. Repeating. Now, we're not necessarily talking about the same kind of palindromes here, right? It's a little more controllable because we can actually design the CRISPR guide in a way that we can have it target specific sequences that we want it to, any sequence that we want it to in the entire imaginable world, which means now we don't have to engineer in sticky ends. And it also means that now if I introduce an enzyme that cuts at this sequence in your body and introduce the fragment that might match with it at the same time, potentially fix your genes that way. So that's the idea. People are doing garage science, I'm not kidding, with CRISPR and trying this out on themselves and trying to insert, like relatively tame genes into their body to make sure it works before they try to cure their child of leukemia, which is usually what drives these people, something like that, right? So before they're doing that, and they'll be like, oh, I got the gene in there and it's showing, fluorescent protein in there, and it's glowing in that area. So now this works for sure, and this is working. Well, if you're just injecting the mRNA or something into your body, yeah, it's going to make it just like the COVID vaccine, right? Yeah, I mean, it's going to deal with that, but it's not going to stay there permanently. And that's the fact of it. It turns out. Give them a few days. Once we have some cell turnover, doesn't stay. So to control all of that stuff and to make it work at specific genes and only the genes that you're trying to target without causing any damage anywhere else in the cell, that has proven nigh impossible. So if you thought CRISPR was a magic wand in science for all medicine, we can now change any gene we want to ever, CRISPR may not be, at least right now, with how our technology works. Right now, it may not be what we're looking for. We have never got it to work in human cells. Some people with conspiracy theories like to hear my conspiracy theories. Some people with conspiracy theories, which isn't one that I believe, but would say that the pharmaceutical companies already know how to work CRISPR and they're just not sharing that information because they want us to need the drugs to treat ourselves. That's fine. We can think that about pharmaceutical companies if we want to, but imagine the bank they could make if they could use that CRISPR technology. So I promise you, if they knew how to use it, we'd know. I promise. Okay. So drop it out of your minds, because they would be milking you for every cent, man. All right, polymerase chain reaction. This is PCR, in case you didn't figure that out from the polymerase chain reaction. You do have to know what PCR stands for. That is something that will be on both the Unit 2 exam as well as the midterm for the lab, because we're going to go over this stuff there as well, because this is lab stuff. All right, so it will increase the amount of DNA in a sample without needing to grow it up in a culture or anything like that. We can just take one DNA molecule, literally one, and multiply up multiple. It's very sensitive. It can detect cancer from only one cell. So if you have one cell in the area that we sampled that happens to be a cancerous cell, as long as we got that one cell, we got you covered. We can tell you whether you have cancer or not. It can also diagnose an infection from one gene copy. One gene copy means one piece of DNA. That means you swab your nose and send it off to DLO to have it tested for the PCR machine. They only need one virus on that swab. So odds are if you're negative, you're negative. Like, it's going to be negative with those. Okay, so it can diagnose an infection from one single gene copy. Very impressive stuff. And this is widely used technology. Let's see. Uses the same events as DNA replication, where we open up our helix. We went through that replication fork, right? Well, they don't really have to do all that mess because we're not trying to do a whole chromosome or something. But we'll open up our DNA, exposing the strands so that we can use them as templates. We still have to add primers, right? Remember how we had to do that with DNA replication? We had to add a little bit of RNA so we could build off of something, because you can't build off of nothing. So they have to have something to attach to. So we'll give them a little RNA primers. And this may be, they sounded like maybe in your replication talk, you were thinking, okay, the primers, I mean, we need them, but they're not that important because we're going to replace them with DNA. In PCR, these may be the most important thing in the entire testing, because this is what's going to allow you to test for the gene you're interested in, instead of just DNA is there in general. Because if I take a swab from your nose and I'm just looking for DNA in general, everybody is going to have DNA in general. So how are we looking for COVID though? And if you're sick and you have a fever and you have a snotty nose and you go to the doctor and they swab you and they send it off for a PCR test to determine if you have influenza or COVID or RSV, for example, they have to know what they're looking at, as opposed to some rando cold virus that nobody cares about, right? Like rhinovirus or something. Which there, by the way, are 200 different viruses that cause the common cold. So how do you separate those three out? So you have to have primers that will sit in specific places on specific genes associated with HIV. So let HIV, well, it could be HIV, honestly, but anything, honestly. We're saying COVID. So COVID spike protein, let's say, the gene that codes for COVID spike protein. Create primers that will only recognize that, or should only recognize that. Our primers here aren't going to be a few nucleotides. Our primers here are typically about like 15 to 30 nucleotides, depending on how specific that sequence is. And there's algorithms for that. And scientists typically design these primers ahead of time, knowing like what would be more useful for targeting blah, blah, blah thing, and then order it from a company that will just make them from the nucleotides. Because making them from scratch from the nucleotides in mass, that part is kind of prohibitive to making making strides in research. So they usually have companies design, make them for them, but the scientists have to design them first. In all research, and I had to design a lot of primers, believe me. So that can tell you specifically if you are binding at a COVID-related gene, or an influenza-related gene, or RSV-related, or HIV-related, or anything. If you have the genetic sequence that you know for that virus's genetic makeup, you can design primers to detect it. If your virus is an RNA virus, you're going to need reverse transcriptase first, because this is only for DNA. So that matters as well. But there's stuff like that, but the concept of this, they're literally making copies of DNA. That's all PCR is. Once we figure out what part of the DNA we're interested in, we're just making copies of that part over and over and over and over. That's all it is. It is not fancy. It really isn't, but we couldn't have done it without a few things. So we know we have our primers, 15 to 30 bases, so that we have specificity for the gene that we're targeting. We have DNA polymerases. Again, you need it, right? It needs to add the nucleotides somehow. So whatever kind of DNA polymerase. However, this whole process is going to be driven, like I said in the beginning, by heat. And if heat denatures the DNA, y'all, guess what else it denatures? The proteins. You guys know what DNA polymerase is, right? It's a protein. Hopefully you understand that enzymes are proteins. They're not a random, like, metal or something sitting out there. So they are proteins. They rely on their hydrogen bonds for their structure and their function as well. So if heat is disrupting the structure of the DNA, it will disrupt the structure of the DNA polymerase as well, the enzyme. So what did we do about that? We found organisms that live at deep thermal vents in like the frickin' Mariana Trench. That's like hot, like 120 degrees Celsius. And we took their DNA polymerase because it has to be adapted for heat, right? If they can survive. So we took theirs. Isn't that kind of cool? Couldn't have done that. PCR exists, this whole process. The #1 most used diagnostic tool in the world exists because we discovered the DNA polymerase of some bacteria in a deep sea vent. Really neat. Anyways, we call it Taq polymerase typically, but PCR happens in 3 steps. and it really is just three. Denaturing, which just means breaking apart the DNA strands. So their natural state is to be double-stranded, or denatural stating. We're going to separate the strands. Then we're going to add on our little primers. So the heat separates, right? And we'll cool it down a bit so that our primers will come in and stick. And again, those should be in excess so that you know that they're sticking on there to get appropriate product, right? Once they're stuck on there, you can have your polymerase start working, and it'll just start doing it on its own, adding nucleotides. So I just told you, all you do is heat to open up the strand, cool it a little bit, the primers will bind, and then your polymerase can just do its thing. You have nucleotides in your little solution, in your little tube that you're putting in what we call a thermocycler, which is the machine that does all of this. And then it'll go over and over and over again to amplify. And that's it. That's all it is. That's all PCR is. And it will go from one strand to two double strands, whatever, for each round. So if you start off with one DNA molecule, after one round, you've got two. All right. This is where you might get tricked up or something, because I have this question on the lab exam for sure, is how many DNA pieces would you have after this many rounds of replication? It's not going to be 94 or something, okay? It's going to be like a low number. Typically, it's like 4 or 5, something like that, okay? But if you have two after the first round, and each one will double, that means each round doubles. That's all. Not that hard. So if one becomes 2 in the first round, in the second round, 2 will become 4. In the third round, 4 will become 8. In the fourth round, 8 will become 16. In the fifth round, 16 becomes 32, and so on and so forth. But you can see how those numbers can ramp up pretty quickly as we're going. So you can get potentially millions of copies made from one DNA strand in a matter of like 40 rounds of replication. And that might take you 2 hours, maybe, if that, depending, right? So tell me that's not amazing. So what we care about that, you can't detect, I promise you, you cannot detect by itself one strand of DNA with anything, ever. Nothing. You can detect one strand of DNA. But you bring that sample into PCR and multiply it, that's a different story. Because now we can multiply it. Additionally, you have a rare sample of a tissue from this person who has this rare genetic defect, and you were able to get a biopsy before they died. Now you can look for that gene that was defected and amplify it. So you can start working on your research to look at ways to treat it for other people so that they don't have to suffer the way that person did. This is so useful in medicine and science. It's just mind-boggling how useful it is. Okay, so I do want to come back to one thing. Remember, though, for you to make your copies, you need the primers. You know we need the primers. And because we're wanting to know if you have COVID, we need to know that you're amplifying COVID DNA. So we have COVID primers in there. So if you go into the doctor and you say you're sick, and he swabs your nose, and he just pops it in a PCR machine, what is that going to do? Nothing. It doesn't mean anything, actually. It doesn't mean anything if you say it to me like that. So I just feel like that doesn't make sense. So where's the missing pieces? You have to know what you're testing for. You have to know what you're testing for. Okay? If you have a patient that you're trying to diagnose, you have no idea what's wrong with them, there's not a PCR for that. You need to test each disease individually. There's not like a special mix that will tell you the answer to anything. Because if there's that many primers in there, they'll just be binding together and mismatching. And then how, if it's positive, it comes out, yep, we have stuff, multiplying in here. How do you know it's COVID versus influenza versus, how would you ever know? You can't. So there are a few tests out there like COVID, influenza, and RSV. You can test for those all at once. If you have the right kind of machine that can deal with it and the right primers so they don't bind with each other. And because, like I said, right kind of machine can tell which one is amplifying as opposed to the other. Otherwise, how would you know which one was amplifying? It doesn't tell you. It doesn't know. It's just amplifying. It's not the smart one, you are. You have to interpret it. So there's these things called real-time PCR, which I used at my old job. Oh my gosh, so much. Real-time, both my old jobs, actually, I used to work, in case you guys don't know, other than the food testing place where I talk about a lot, but before that I worked at Integris Baptist's, what am I trying to say, transplant testing lab. There's only like 6 people in that lab testing for anybody in pretty much the entire region that's getting a transplant. And if you also didn't know this, Integris is the only hospital in the state that offers certain kinds of transplants. You didn't, huh? They just started their heart transplant program, y'all, at OU. Just started it. Just to be clear. Yeah, yes. So I want to be clear, yes. I want to be clear, like they only did livers prior to that. And livers are baby talk. So like, it's a big deal. So we did all the testing for anybody that was getting any organ, basically heart, lung, kidney. We didn't even do liver because it was a waste of our time. We didn't even care. That's how baby a liver is. We don't care. You don't have to match it as specifically. And you can take it from a living person. All I have are these organs from this brain dead person being shipped to me from Atlanta, you know, in a freaking helicopter. And you get these samples to work with to determine if it's a match to these people that are on the next in line at your hospital from UNOS. You better not. They had to come to Integris, like patients would have to come to Integris. So they would, they might get flown if they were in a bad enough shape.
Well, my son got a heart transplant.
Yeah, so it doesn't, it matters like what hospital you're closest to or what's, you know, better available for you. So the region, I mean, more or less, like, I don't even know if Tulsa does heart transplants. I don't think they do. Yeah, I think it's the state of Oklahoma, and each state I think has their own like control over that sort of stuff. But
like St. Louis was heart and he has a heart transplant and then
yeah. So there's just, it's not like really, people think you can just go to your hospital and get a transplant. Nope. There's like one in each state that you can go to. And Integris is that for us. So I got the opportunity to work in their lab. Anyways, we had to, before you could even be on the list for UNOS to get your transplant, we had to sequence your DNA. And I don't mean the whole thing. We had to test you for specific immune markers that would be relational to every single immune marker that we knew of in all of science, that we were relational to possibly rejecting an organ. So we do your DNA to try to predict that. We would also test everybody for any possible antibody interaction that science had knowledge of. This is a lot of work to get somebody in, and usually months of work before you can even officially get put on a list for UNOS. And then once you get put on the list for UNOS, now you wait for the right person to die. Because you don't get lungs from a living person. And you don't get lungs from a dead person either. These people have to be brain dead only. And they have to be organ donors. You're starting to see how rare it is to get the right organ for the right person at the right time. Very uncommon. A lot of people die waiting. And all this depends on the genetics behind it all. And that, of course, and also living organs, but all of that genetics behind it all is done with PCR, all of it. So we do PCR to amplify the gene. And this thing, like I'm trying to tell you about, which is called real-time PCR, you can have your A associated with a red fluorescent color. Okay, a red fluorescence, it glows red whenever it gets bound onto the DNA. T, it glows green when it binds onto the DNA. C, it glows blue when it binds on, you know, whatever. Over the different colors, should have 4 different colors, right? For A, T, C, and G. And the machine can detect every A, T, C, or G being added on as you're multiplying on each round. And so maybe the first round you don't have enough for it to really say definitively, but after like 30 minutes, you can look at your curves of what colors to see what's getting added on. Because if stuff is getting added on repeatedly, that's a positive. It has to be. because you don't add stuff on when you're negative, because you shouldn't abound with those primers. You should have nothing if you're negative. It's nothing or everything. That's how PCR works. But you can see it in real time while it's happening. People, man, science is so ridiculous. So anyway, so at my job, we knew if something was going to be positive before it was done. We had, by protocol, as all hospitals would as well, you have to run it to the end of the procedure, but you get an idea before, well before it's done running. And the ability to do that, it's kind of, sensational in a way. Okay, so we can move on because I don't have any time. I always do this, like the first few slides I get so freaking backed up on. Okay, recombinant DNA, because is it not exciting, right? I feel like it's just so cool. Making cloning vectors. If you want to use your bacteria to make your insulin, right? You'll have your genetic donor, maybe me with my pancreas that makes insulin. That'd be a genetic donor. Then we're going to insert my gene for insulin into a vector, which might be a plasmid, which we've already learned about. It could also be a virus that's going to insert its DNA into the thing's genome, in this case, bacteria's genome. And then insert that vector, whatever it was, into a cloning host. In this case, we were talking about bacteria cranking out that insulin. So that's how cloning works. We can already make the complementary DNA from mRNA transcripts and then amplifying by PCR. We already talked about what plasmids are, bacteriophages, we know what they are, right? Bacteria get infected by viruses and then they can potentially insert the viral DNA as prophage into their bacterial host DNA. This is an example of a plasmid. You can see the brown at the left, AMP. You can see the yellow at the bottom, ORI. That's just the origin of replication, where if this was going to replicate inside of a cell, that's where it would start from for its replication. That's all that means. This is called PUC19, a very commonly used plasmid. And ampicillin is on there. That's because once you insert your gene into whatever site you chose, let's say you were using EcoR1, that matches up with what you were engineering on your gene. And that's what you're going to insert here. Hey, you found a plasmid that has an EcoR1 site. So you can order plasmids that have the site that you engineered in there. The one that matches better what you're trying to do. Anyways, cut it with the EcoR1. Have your sample cut with EcoR1 and mix them together and stick them in there. You see how those things are found in the middle of that light blue arrow? That light blue arrow is a gene. And so if you're inserting it correctly, you should interrupt that gene. And so that can also tell you if you inserted it correctly, number one. Number two, whether your plasmid got into your bacteria or not, you will plate your bacteria onto media that has ampicillin. So only the bacteria that took up the plasmid should be ampicillin resistant. So the ones that took up the plasmid are the only ones that are going to grow. You see already how just efficient that is. And they're all designed to be like that. And we'll learn more about that in lab. But that's transformation typically that we do that. So we can use this process to make all sorts of things for humans, not just, one of the first things was insulin, but we can use this from mRNA transcripts from all sorts of stuff to have cells cranking out whatever protein we want them to. All right, so we already learned about the cloning vectors and we learned about cutting and ligating back together. Then we stick this into our cell. It's going to go through transcription and translation, crank out protein into its medium, and we can purify that protein from that and shove it into a vial so you can inject it for insulin or whatever. All right, there's something called synthetic biology, which is creating cells from scratch, which is a real life thing that has happened. In 2010, using just A, T, C, and G, and just the understanding of how cells work in general, they just used A, T, C, and G to make up DNA that then, and just giving it the resources that it needed, the nucleotides and amino acids and stuff, they made a cell all on their own. I don't want to know about it. I don't want to know. I feel like that. I don't know how I feel about this. I don't want to know. But yeah, so that is synthetic. That's completely synthetic, creating life from scratch, just with the little resources that we have that wasn't life before. All right, we can obviously use a lot of this to create maps of our genome and sequence, use random primers to do PCR, and whatever the products are, we get those and sequence them. And then from that sequence, the next one, oh, okay, this one lines up with this one. Oh, hey, this sequence matches up with this one. Oh, and this one matched up with this one. Suddenly you're building a whole genome from that. And it takes a long time, but you can build entire genomes with just random primers or designing from like the first set of random primers. That's how that we have mapped out the entire human genome, but we don't have every single possible allele, which is different versions of your gene, mapped yet. We just don't have all of those available yet, haven't been sequenced. We know what area codes for what gene, but not all the different kinds that could be there. So cool though, right? So we can create maps of entire genomes and stuff. In order to do a lot of this work, we need to go through gel electrophoresis. Gel electrophoresis is kind of straightforward. I feel like it is, but maybe not to you guys. We're going to make a gel that is clear, and it's made from agarose, which is a sugar that comes from, guess where? Agar, right? No biggie there. So it's going to have that sort of jelly-like consistency like our plates do or your slants do. And it's clearish. And you'll get your DNA samples, and you can put them in there with little special stains or special marker or whatever it is, so you can see it later, because you don't want to just be looking at a clear gel. And then pop it into some liquid just to keep everything happy and nice, because you're going to shove electricity through that whole thing. And because we already know DNA is the backbone, right? If we look at the backbone of DNA, sugar, phosphate, sugar, phosphate, sugar, phosphate, over and over and over again. What charge does phosphate have? Negative. So DNA has a negative charge. So because of that, it'll move to the positive end of the gel. Maybe just moving your sample through the gel doesn't sound useful, except that this gel is very finely, milled or whatever. So the small molecules can move just fine through the gel, but the big ones will get trapped up. So like a big old webbing or something like that. The bigger the molecules will move slower, stay towards the top where they started. The smaller ones will move faster through towards the positive end. So now we're separating out those sizes. So now when I tell you guys that I can take this tiny little drop of blood up here that only has a couple pieces of DNA, look for a gene that humans have in general, amplify it in PCR, and take blood samples from all these people in the class. And then, after we amplified it on PCR, cut it with our restriction enzyme at whatever places you want to cut it, right? EcoR1 it, everybody gets that. And now run them out on this gel. And now we're going to see those different sizes of fragments that we can get. Those restriction, fragment, length, polymorphisms, we can see those, the differences between people, okay? So that's what we can do with gel electrophoresis. It also allows us to take a sample from somebody and like stretch it out, and we know that this gene is blah, blah, blah length. So you can cut that out of the gel and literally purify the DNA out of the gel. So you get your target gene exactly what you want. So it's useful. But DNA moves, it's negative, it moves to the positive. The smaller ones move faster. And you can already see, if I were to compare you, look at all these different people and how the different ways that they have different bands on here and stuff like that. Even like some that are kind of similar, like 5 looks almost like it would have been similar to 7, but there's got a few little differences, right? That might not be there. So stuff like that already tells you differences between people. Not to mention, you can see, what is it, 3 and 4 here? Look at 3 and 4, man. That's the same person, y'all. That's not your sister. That's the same person. That's how specific this is. Twins can have the same, identical twins, obviously, right? But outside of that, everybody different, okay? So cool, we can use that. We can also use sequences of DNA to just match up and match up. You know how the primers have to match up with the sequence that you're looking at, right? Well, you can use that concept just to see if there's any matching DNA in your sample. So we can make you say, this gene, this probe I'm making, it looks for, this defect in, let's say, breast cancer, the BRCA gene, BRCA1 gene. BRCA1 gene is associated with high incidence of breast cancer if you have it in your family, especially if you have 2 copies. So if we were to take your cells and have a probe that is fluorescent and mix it with your cells, and that probe only looks for cancer-associated BRCA genes, bind the DNA specifically, And whether or not it is going to be fluorescent or not, or whatever marker you use to identify it, will tell you if it's there or not. So you don't necessarily have to go through all these processes of sequencing and everything. You can do this first and then decide if you want to go into more detail with it. So that is useful there. So that's nucleic acid hybridization and gene probing. You can also do these gene probes and nucleic acid things in situ, which means looking at like live tissue. This is just a picture depicting it. If you want a better explanation than what I'm trying to give, you can see this is just with like bacteria, showing you how they would do it with bacteria, but you can do this with actual people. They do this with actual people. Okay. So I just want to get to like the big stuff. Sizes of DNA, this is something I don't want you guys to miss. We write down our size of our DNA based on the base pairs that are in your DNA sequence. So your base pair, like if I were to say, oh, this person's gene reads AAGCC or whatever, each of those is a base. But AAGC has an opposite strand, right? So it's actually whatever the complement is to it, but it's two strands. So that's a base pair. So when we say AGCG, we're really talking about the double strands. It's a base pair. We refer to these as base pairs and we count them by the number of base pairs. And the size of DNA is determined according to that. And kilobase pairs is going to be with a K in front of it. So BP is base pairs. Kilobase pairs or kilobases, which is also just the B, either way, is 1,000. And then, so that would be 1,000 of that. And then a million is mega. And those are really the only ones you have to know. Regular base pairs is just singles. Kilo, 1,000, and mega is millions. Because I'm going to have you guys on exams. I think it's YouTube and the lab, but it's definitely on the lab. I'll give you some numbers of sizes of DNA listed in base pairs and kilobase pairs and megabase pairs, and you're going to have to tell me which one is the smallest or which one is the biggest or something like that. Okay. People often get it wrong because they just look at the number. They don't look at the kilo or anything like that. So we talked a little bit about these things. So we don't need to go through this. Using those fragments to identify killers and things like that. And creating sequence maps. We did talk about this, random sequencing, and then building off of that to create genomes. A study of this stuff is called genomics and bioinformatics. Just FYI, I don't know if anybody cares, but there you go. Okay, so annotating the genome is just looking at the genes that are associated with different proteins that we might be concerned with, which is helpful when we're diagnosing diseases. We also have something called single nucleotide polymorphisms, which we wouldn't be able to identify if we didn't do this, right? So single nucleotide polymorphism just means that from one nucleotide change, your protein product is different. So let's say you had AAA and it got changed to AAU, and you had phenylalanine, which is a particularly hydrophobic type of amino acid, but it got replaced with glutamate, which is charged. Well, that protein is going to be folded completely different from just that one amino acid change, right? From that one nucleotide change that led to that amino acid change leads to that protein change, and that leads to disease. That's a single nucleotide polymorphism. Some of them, we're talking about diseases. Some of them, we're just talking about maybe different color of hair, different color of eye, or something like that. But just one nucleotide change in a protein can change everything. One nucleotide. And there's studies out there looking at the importance of these and looking at single nucleotide polymorphisms, the possibility of changing one nucleotide and causing a different outcome in an individual. 10 million of them in our genome, in the human genome, not in your genome, in the human genome. I don't know why that happens. I don't have a good enough explanation for that, but no, this isn't something that changes in you. I'm talking about like amongst all people. Yeah. So you might have a version for, blue eyes, and then somebody else had one nucleotide change in there that gave a different protein, and now they have, blue-green eyes or something like that. It's completely different color. That's what I'm talking about. Yeah. So that's a single nucleotide polymorphism. There are 10 million different versions of these possible single nucleotide changes in the human race. Makes sense. So if you have thrombophilia, it's a single point mutation, one nucleotide change, and that will have increased clotting and put you at risk for strokes and things like that. So that's pretty serious. One nucleotide. Okay. So this is just talking about more detail into that if you're interested on how a nucleotide can change into a different amino acid, which can change into a non-functional or poorly functioning protein and lead to disease. That's just a picture version of it. All right, microarray. Remember how I was telling you guys about how you can take this probe and then see, oh, this matches up with this person's, breast cancer gene, and this person has it in that tissue, right? We can take on the other end of it an entire plate that's dotted with all of these basically probes. DNA probes, little tiny ones, for different diseases or different kinds of diseases, different versions of the disease, different strains of staph, different strains of E. coli, whatever. 365 different samples on one plate, put the patient's sample on top of that whole plate, put it in a machine and see which spots flare up. And you can say this person has MRSA, not this kind of staph, or this person has a worse version of gonorrhea or something like that. So that's a microarray. Obviously pretty useful, and we can get down to subtypes of cancer, so we can get better treatment of cancer by doing that. So it's not only going to be infectious diseases. Okay, if I need to tell you what we have done with recombinant DNA technology, I don't know how to help you at this point. So, okay, so one of the things too, and I just want to touch on this because I know we're running over time, but things like Remicade and Humira, we will come back to these in the immunology chapter, but adalimumab, right? That's Humira, yeah? Then there's other ones like blah, 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 blah mab and blah, 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 blah mab and whatever, right? If it has a mab at the end, that means monoclonal antibody. We're going to talk about what antibodies are and what they do, but that's a human monoclonal antibody protein. We can put that gene for that protein into bacteria and they'll make out that antibody. So why do we want an antibody? If you have an inflammatory reaction to something, you have signals sent out by your cells called chemokines or cytokines. And those cytokines will signal for inflammation, let's say. And you can have an antibody that you can design to block on the receptor. So if you are signaling for inflammation and it comes in, right, and this causes inflammation in your cells, it triggers the whole thing. Adalimumab and other similar things like Humira or Remicade, the antibody comes onto that receptor and you can't communicate that signal anymore. So you can't have inflammation. So blocking inflammation by using that. I'm sure you've heard if you take either of these drugs or any similar drug, it decreases your immune function because inflammation exists for a reason. And they've proven that with that. You're supposed to have inflammation, right? But anyways, that's how they work. That's why you are also reduced immune function if you're on it. These are just some of the things that they've used. Genetically modified organisms. Okay. Use all this stuff to create. Genetically modified organisms. Okay, Jesus. Again, this is another one that I just want to touch on. But this is, let me see what else. Is there anything else that I give a crap? Gene therapy. Yes. Okay. All right. GMOs stands for genetically modified organisms. They're also called transgenic organisms. That's the same term. Okay. Any organism that has ever had any gene put into it that wasn't there naturally is a genetically modified organism. So these bacteria that are cranking out the insulin, genetically modified organisms, right? Guaranteed, any corn you've eaten in your freaking probably last 10 years is genetically modified. Guaranteed. 98, I think now, % of corn in the United States has this gene. Oh, it's not this gene. It's in the next slide, right? Has this gene in it, which is a gene that allows this bacteria, Bacillus thuringiensis, which we already know is an endospore forming, right? Anyways, it creates a substance that makes it so, it staves off pests. Let's just leave it at that. But anyways, insects, they take the protein from the bacteria, put it into the actual corn's genes, and now it can't get insects eating it. And hey, yay, now you get corn that isn't treated with pesticides. And that sounds great. Except that plants, I want to be clear about this, plants, not y'all, plants can have the possibility through transposons, which allow them to move their genes to wherever they want to, basically jumping genes is what we call them, to other plants that maybe shouldn't have that gene, that we didn't put it in. So they can transmit, it's a lot more complicated than that, but they can transmit those genes for pesticides, for pest resistance to weeds, to other plants that might not be in the area potentially. That's a big problem with GMOs. They didn't think about the environment and how they were affecting the environment. Of course not, right? So I don't know what to tell you about that. If you are of the mindset that you can eat a genetically modified organism, or you've been told by somebody that when you eat a genetically modified organism, that you can eat it and get these mutated genes in your body, I want to seriously ask you to look at that again. Because we have already talked about, we'll talk about metabolism as well. When you eat something, you don't conserve its DNA sequence and save that for later. Why would you do that preferentially for GMO versus any other DNA sequence? So what makes that special though, that you would keep that one? First of all. Second of all, when you eat chicken, let's say, and you break it down in your stomach and whatever, you're going to break it down to the nucleotide level, and your body will use those nucleotides potentially, or the parts of the nucleotides to make more things in the body. But at that point, it's unidentifiable as GMO, right? So that's what I want to say. Impossible to get those genes in your body from eating a GMO. Come at me. Seriously, like this is impossible. I don't care who's told you. It's your mom or your aunt, or if you read it online from your favorite Donald Trump website. I don't care. I'm telling you, it is literally no. Okay, now that we cleared that up. The more you learn from this class, I'd hope that you would agree with that. You don't need me to tell you. Last thing, gene therapy. We can take a virus like HIV, put a new gene that works into it, like the gene to make insulin properly, or like a gene, for all these defects that could potentially kill children or whatever that can be heritable. And we can put that into HIV, clear it out, make HIV carry that gene for us. Instead of inserting its genes into your cells, it'll insert that working gene for that protein into your cells. And we can hijack that mechanism to have the virus then go around inserting your target gene into cells of people that don't have a working version of that gene. This is real and it does work. We've helped treat 13 different diseases so far with this sort of technology. You take your bone marrow out and slush it up with these viruses that we've designed, let them be infected, pop it back into you through a little IV infuser, and now you've got cells that are infected in your body that can disseminate it throughout other cells in your body. And it works. This is called somatic gene therapy. That is going to be only affecting your body cells. Germline therapy, now we're going to be treating the egg and the sperm, early embryos, and things like that, so we can actually make it heritable to future generations. But it doesn't exist yet. So only the somatic ones do. But cool, yeah? Okay, I think that's it. We have small RNAs, I don't really care that much. Yeah, that's it. I don't think I have small RNAs on the thing, but anyways, we'll be just matching sequences to the DNA that help block the DNA sequence. But yeah, I always forget how long I take this. Like a short lecture, it looks like it's gonna be short, and I always get carried away with the PCR. I literally go into every class thinking that this time it's gonna be short. Never is. Last time we got carried away talking about the healthcare system and universal healthcare and stuff. I didn't start the conversation, to be clear. But people usually get interested in this stuff. Oh, sign out.